Okay, good afternoon, everyone. Let's start. Let's start the class. And a quick announcement. I hope you are all doing the assignment, and it's a graded one. So I hope you're all doing it well. Yeah. Anything after the deadline will have serious penalties in terms of the grade points you would receive. Okay. If the audio is not clear, please do let me know. If it is clear, somebody let me know. Then I know that it's not the something wrong from my side. So, in the previous classes, we have learned some things. We did discuss about R and or the discovery of restriction endonucleases. We learned about what are R and M systems, restriction and modification systems. While we did that, we also learned why we may have restriction plus modification, and we may have on the genomes we can have these two as a combination. We can have this as a combination, that is restriction modification is mutated, but methylation or modification is fine. We may also have R minus and M minus. Both restriction and modification are not present. That is fine. But it is unlikely that we would ever have any genome would have restriction is working, but modification gene is mutated. This situation you will never be able to isolate, because we know that methylation provides immunity to the genome from being broken down or degraded by the restriction endonuclease. Okay, so we also learned about the nomenclature, which we will again have a look at it. And this is the first alphabet is capital, that indicates the genus. Second two alphabets are indicative of the species, and this one indicates the first enzyme or fifth enzyme. Here it is the first enzyme. So the organism names always should be in italics. Either you write in your text or in restriction endonucleases as a nomenclature, you should always use italics. Okay. And in some cases you may have, we also were discussing about, let's take HIND3 and BAMH1. HIND3 is for Haemophilus influenza, and D is the strain name. Some of them do not have strain name. Here, as in the case of SMA1, there is no strain name, so it is not included. But others may have a strain name, like here it is D is the strain, so it is put as that. D is not put in italics. And then we have 3 Roman numbers, which indicates that it's the third enzyme isolated from that particular organism. And if there was another one discovered from the same organism and strain, then it would have been, it would be HIND4, something like that. Here you have BAM in italics, and B is capital and AM is small, but H here, the strain name is capital, because the strain designation here is capital. That's why it is capital. Okay. Yes. And we learned that there are many different types of restriction endonucleases, and some of them are 4 base cutters. That means their recognition sequence includes about 4 bases. While I said this, I'll just strictly discuss this. So there are, for every restriction endonucleases, there are 2 properties. One is restriction or endonuclease. That is, it is going to break the DNA or break the phosphate backbone. And there is one more, that is sequence specificity. They do not go and cleave anywhere randomly. They are going to cleave at particular place. And out of the different types of TA systems, we we realized, or we know that now, that type 2 are most preferred for several reasons. One is that R and M are on different proteins, so you can just use this one. Secondly, it doesn't require so many cofactors. And third important or most important one is they recognize the specificity and endonuclease activity are on the same site. And that's why type 2 restriction endonucleases are mostly useful for genetic engineering or construction of recombinant DNA. So we have 4 base cutters, and there are 6 base cutters. These are the predominantly used ones, and there are also 8 base cutters. That means they recognize about 8 bases. And I think we can already say that the regions are usually palindromic, in the sense that you read from 5' to 3' in either of the directions. It sounds the same. GAA-TTC. If I have to read this, GAA-TTC. I will tell you this once more. I already repeated that. So, and this is one sequence, right? And there is another sequence. I will write this. These two sequences are not the same. Okay. Always reading should be from 5' to 3'. And so GAA-TTC. And if I have to read this one, you should never read from GAA-TTC. That is wrong. It should have been CTT-AAG. Okay, so these two are not equal. So keep that in mind whenever you're trying to identify restriction sites and so on, especially when you're also we will do an exercise on this. An assignment will come up in a week or so. At that time you will be able to do that. So why should it be palindromic is something. This is a nice picture of it. What you're shown here is the DNA, and the restriction endonucleases are kind of dimeric, homodimers. If not all, at least many of them are homodimers. So this is the ones shown in cyan is one kind of one subunit, subunit, and the other one is they're shown in green is the other subunit. They have this active site, and they both are homodimers. So the sequence recognizing will also be similar, and that's why, and that is where they usually make 2 cuts. One cut on one strand and the other cut on the other strand. We don't need to bother about it too much, but it is just for understanding's sake. So there is always an axis of symmetry, and that is where we said we'll have a palindrome, and reading direction is this way. And for the complementary strand, we have to read from 5' to 3' as shown here. Then there is also one more thing is, as I said, remembering this much can be easy enough to get the rest of it for the same reason, because it is palindrome. There is another way of another additional factor that is shown is there's a slash here indicated, and there is an asterisk here. Slash indicates the site of phosphate backbone or nucleus. Where would the cut be made? A cut would be made in these places. And asterisk is the place where modification is usually done. That means here this adenine is going to be modified. And when this adenine is going to be modified, the restriction endonuclease cannot cut here. Right? It's not the same with all the cases, but we will see about it. I hope you understood what does the slash stand for and what the asterisk here stands for. There are variations to these based on different types of restriction endonucleases. I think we all know. We will take the same thing as the sequence that is given, GAA TTC, and the slash is here, right? This is the slash. That means the cut site. And I will put this so that you will not be confused. Sorry. 5 prime GAA-TTC. This is the 3 prime, and this is the cut site. If DNA was cut in that location, we would have had G and then C, TT, AA, and the other strand would be AA, TTC, and this would be the G. Right? So anytime, so the ends of this one, this end would be 3 prime, this end would be 5 prime, this is 3, and this is 5 prime. So this region here is referred to as the overhang, because this is the part, if you can see it this way, this part region is extra single-stranded region that is there. So you call them, refer to it as overhang. And there are two different types of. They're also called as sticky ends. We will see more about it later. Because they're called sticky ends because this this T can base pair with this one, and they're absolutely complementary regions. If we let it, they might have a sequence something like this. I'll just put it like full length. Let me do that. And something like this. So here would be AA, TT, this is C, G, and here you will have AA, TT, and C, right? This is the G, 5 prime and 3 prime, so on. So why I left this space here is to indicate that there was a phosphate backbone breakage. Okay, the space here, the discontinuity that I'm putting here is the discontinuity in the phosphate backbone. So they still can be held together by these hydrogen bonds between these two. Okay, there are about 8 hydrogen bonds, so they can be held together for a while based on the conditions, of course. Because they can base pair like this, they are referred to as sticky ends. Okay, and there can be sticky ends can be 3 prime overhangs or 5 prime overhangs. So now this one is having a 5 prime, and this is the overhang, and it is 5 prime. So the one that we are working with is having a 5 prime overhang. And what are blunt ends or flush ends? Is if there is a, we will see in the other ones. For simplicity, I will take if there was a restriction site, something like this, and if it were cut within the axis of symmetry, or not necessarily, yeah, it's always usually it is the axis of this on the axis of the symmetry. Then you will be these will be GGG, CCC, and the other one will be CCC and GGG. So these are called as blunt ends. There is no overhang or no sticky ends. So those are called as blunt ends. And this is what I have already described, just showing it. Only thing I should add is the this one is the 5 prime overhang, and if you see a different for PST1, CTG and CAG, that's the site, and its point of cleavage is here and here, because of which now you have a overhang, and that is a 3 prime overhang. This is 5 prime overhang, and the second one, PST1, will generate a 3 prime overhang. As is described for a previous one, SMA1 recognizes CCC, GGG, and it cleaves at the site of axis of symmetry, because of which you will have two different DNA molecules with no overhangs, and those are referred to as the blunt ends. You can also call the other ones, for the ones generated by PSD1, as 3' sticky ends, and by EcoR1 as 5' sticky ends. So I want to go through this exercise, the one that I'm showing here. This is about, we'll use some of the 4-base, 6-base, and 8-base cutters. What does this generate? What kind of overhang would it generate? 5' or 3'? That's what you should try to figure out for all of these. Can you first tell me which ones will make? You can all answer as you know the questions. So MBO1, DPN1, and SAU3A1, they cut at this. They recognize a 4-base cutter, and they cut here. What kind of overhangs will be generated? 5' or 3'? Hello, you're supposed to answer, so I'm waiting.
3' overhang, sir.
How is it 3' overhang? This is 5'.
The cutters before the.
Wait, I'll just let me write it so that we'll have clarity. GATC, and this is 3', and this is 5'. The cleavage is going to be here and here, right? And upon separation, what will we get? We will have something like this, CTAG, and here we'll have
it's a 5'.
DC. That's right. So yeah, that's all right. What we are going to always, the first thing is to work it out. So as we work out, we will get more clarity, and that will be easier. So MBO1, DPN1, or SAU3A, they generate a 5' overhang. What does MSP1, HP2? Yes, a different person can answer. Each one, that way we'll try to engage many.
5' overhang.
Yeah, this will also be 5' overhang. And ALU1, anybody else? I think I have to call out names probably. Shivdarini?
Sure.
Yeah. Which overhang, what kind of overhang will be found by ALU1?
It's 5' overhang, sir.
It is cutting at the axis of symmetry. So it will be blunt ends. Okay?
Yes, sir. Yes.
So is the case with HAE3. It is going to be blunt ends. And ACGT will generate 3' overhangs. So, and I hope you now see that based on how you look at slash, if the slash is towards the 5' overhang, this is the 5' and 3'. If it is on the 5' side of the axis, then it's going to generate 5' overhangs. If the slash is within the axis of symmetry, then it is blunt ends. And if it is towards the 3' side, then you'll get 3' overhang. This is just for a quick kind of mug-up, but it's always easy and better to work it out like this I've shown here, so that it will be error-free. Say for example, if you're trying to remember anything like I said, if the cut site is towards the 5', it is 5' overhang. I mean, that's easy. There is no confusion there to remember, to get confused. There is no point there. But if you miss it, then it's going to go wrong. Anytime we are trying to remember something, remember how to work it out. And that gives a lot more capacity, because you're remembering a principle instead of a fact. From principle, you can remember many things, and you can apply the principle to many other relevant places. A fact that you learned, unless that fact is applicable to that particular thing, you cannot use it. So I pay more attention to principles than direct facts as such. That will improve. I think it improves your thinking more. Anyway, I hope you understood the stuff. You can do the same thing with the other ones as well. Okay. So there are something called as isosygomers, neosygomers, and there is also isocardamers. I don't know if I have it here, but anyway. So isosygomers, they recognize the same sequence and they cut in the same sequence. Here is an example, ACC 651 and KPN1. They recognize GGT ACC. This KPN1 also recognizes GGT ACC. And they cut, this is an example for a different one, that is neosygomers. Okay, I'm sorry, I made a mistake. Did I make a mistake here? 1 second, let me check it out. We need to check this. Okay, right now I will just hang around with this neosygomers, isosygomers. Some people even say neo-isosygomers also, and that is going to be a little bit difficult to understand at this stage, I think. So I will stop. I will not continue with this part. But what I want you to understand is, if there is enzyme A and there is enzyme B, restriction enzymes, and one is cutting GAA TTC, and so is the case with this one, and the other one also has the exact sequence, and they are going to also cut in a very similar place like this. What is the difference then? The possibility is that first one is that they both belong to they both are probably the same enzyme. Why are they named differently? They are named differently because this one has come from organism B and this one has been isolated from organism A. So what if the restriction endonucleases or rDNA systems also are thought to have transferred through horizontal gene transfer? I'll try to make space here. So what is horizontal gene transfer? You have a bacterium, say I'm calling it as A, and there is on the genome you have the rDNA system here, and there is another bacterium that is B, and through horizontal gene transfer there are three mechanisms. Horizontal gene transfer is through transformation, transduction, or conjugation. In any of these cases, this particular restriction endonuclease is now moved or it transferred from organism or bacterium A to bacterium B. There are two different species. Okay. Now, somebody has isolated the restriction endonuclease from here, and then we are likely to call it as based on the name of the organism here, and we will call it as ARE just for ease. Okay, ARE. From strain and restriction endonuclease. Somebody else might have isolated from here, and now it will be BRE. They both are actually kind of homologs. They recognize the same sequence, they cut the same, their activity, and even the there would be a lot of similarity with the at the level of protein sequence as well. Only difference is they are isolated from two different organisms, because of which their names are different, right? Here it is ARE and here it is BRE. Therefore, in most cases, isosygomers, I think if I will try to recollect it later, they recognize and cut the same location. Okay, that is how one, that's the molecular basis of or genetic basis of isosygomers. There is one more point that I would want to depend is we will do a problem on this at a later stage when we are trying to use this for studying epigenetics. There are three different types based on the methylation dependency. So as I said before, R and M. M is for modification, and often the modification is methylation. So methylation provides safety from restriction digestion, right? That is what the purpose of it is there. But there are some restriction modification system where there are variations to what we are actually trying to discuss. First one is you can call it as methylation sensitive. That means if a restriction site is methylated, then that restriction enzyme cannot act. And there are some other which are referred to as methylation insensitive, which means they don't care if a sequence is methylated or unmethylated. They will just go on cleave. And there are little more critical things we need to learn about it, but we will see it in the when we are trying to solve the problems. And there is one more aspect here. Arval, you have a question?
No, sir.
Okay, yeah. And there is another one called as methylation dependent, which means it can cleave only if there is a methylation. It can cleave this one, but it cannot cleave unmethylated DNA. So we will try to remember these things. There are three different types of restriction endonucleases based on the methylation dependency. Most of them are methylation sensitive. They cannot cleave in their target sequence if it is methylated. In some cases, they are methylation insensitive. They can cleave the DNA, the recognition site, irrespective of whether it is methylated or unmethylated. And there are some which are methylation dependent. They cleave only if it is methylated, but otherwise not. So I want to talk on this one. HPA2 and MSP1 are isoschizomeres. Their recognition sequence is CCGG. They are 4-base cutter. And there is a problem because I was talking about sensitive and insensitive, right? C is the methylation point. This is first C and this one is second C. You will have the other ones on the complementary strand also, first and second. If the second C of the sequence is methylated, MSP can cleave on it, but HPA2 cannot. Okay, so these kinds of little bit of variations are also there. I think you know the probability of sequences. Say if it is 4-base cutter, how often do you see? I'm putting position 1, 2, 3, 4, and so on. So any of these could be. I think you all have done probability, so it would be easier. In this position, there are 4 different types of bases: A, T, G, C. Any of them could be there. And in second position, the same thing, and same, so on. You would get about something like 64, and then what would it be? Something like 256. I'm assuming that. And so if you had a 4-base cutter, one would expect the particular site every 256 base pairs. That means if you had a long DNA, the probability of that particular site, say for example GATC, the probability of that site in that location is it is possible to be found every 256 base pairs, right? The distance approximate, okay? It's not it's just a theoretical way of doing things. If it were 6 base cutters, you can add more 4. This is, say for example, another one, 4 and 4. That would be around 4,000 something, 4096 or so. That means it would be able to cut the same length of in the DNA, that long, will be cut. The distance would be about 4096 or so, 4 kb approximately. So one can see that the base cutter, the number of bases recognized is inversely proportional to the frequency. Right? If you have smaller 4 base, you're finding it highly frequent. If you have 6, less frequent. If you have 8, it will be much more, it is almost rare. Okay, so that's what I meant by, let me check it again. Number of bases recognized is inversely proportional to frequency. Okay, and you can also, if you want to add the length of the fragment size, approximate, and is directly proportional to the approximate and also inversely proportional, right? Smaller the number of bases is small, then the fragment size is also smaller. Larger, and this one is large, so it is directly proportional. Number of bases recognized is directly proportional to the size of the fragment. Just, it's a way of trying to remember things. One thing we have to understand here is, say this is a good one, average fragment size, they're saying, and produced by different enzymes with DNA from different sources. So DNA is isolated from Arabidopsis, nematode, Drosophila, E. coli, and human. If it is cut with APA1, the sequence is something GGG, CCC. In Arabidopsis, it is 25,000. That's the average fragment size. Whereas in human, it's just 2,000. There is 10-fold difference in it. And can anybody try to answer why this is the reason? You can turn on your, I hope you understood the question. Here, this is the average fragment size. One would expect, say, for example, if it is 6 bases, we would expect that it is about 4096 based on what we are discussing. But the average fragment size for Arabidopsis is 25,000, and human it is just 2,000. The reason is because of the, what do you call that? GC content is one thing. If you have high GC content, and because this particular target sequence is high GC content, one is likely to have more of these cut sites. And there is one more thing that you have to think of. The theoretical values that we have come about should have GC content should be 50%. These values should be right true only if these are the conditions. GC content is 50, which means that all these bases are equal in proportion. And then second point is that there should be random distribution. Not like all the G's in one place. If there is a whole genome, all GC are here and AT are here, then it doesn't work out that way. And the ones that we are discussing here in the beginning is just the theoretical ones. But for solving the problems, we assume often this is a question I would want you to try to answer, and always you should assume, unless mentioned otherwise, 50% GC content is what I mentioned. Assuming a DNA molecule with 50% GC content and a random distribution of the 4 bases, a circular DNA. So anytime you have a question that is coming up, draw the diagrams, think on paper all the time. That is always the best thing. Um, a circular DNA of 2048. So we'll just put it 48 base pairs. What is the approximate number of fragments that could form by cleavage of this DNA with a 4-base cutter? I didn't mention the any sequence, so it should be all right to do that. Why I'm saying that is, if I had a APA, it doesn't have all the bases. It only has GGG, CCC. That means it's not having all the bases in the genome is not going to be of helpful. The calculation directly is difficult. But in this case, I just mentioned a 4-base cutter, so it is much straightforward, and there is no conflict, and no argument can be raised. It's just theoretical, everything. So if you have a 4-base cutter, the size is about, what did I say? The size is 256, I think. The average size is 256 base pairs for a 4-base cutter. And I would, the length is given as 2048, and by 256, it will be number of fragments it should be cut. Right? Anybody can say what is the answer now? It is somewhere around, I think somebody got it right. So B must be the answer for it. Again, for those, if you have confusion, it is a 4-base cutter, provided this theoretically, provided 50% GC content and random distribution, then it would cut approximately. The probability of it obtaining is every 256 base pairs. And I have taken the total length of the DNA and then cut it by 256. And the question is about how many number of fragments it would generate. And from the options, this is the most reasonable answer that could come up. And therefore, I chose that. I hope you understood it. And yeah, please remember that anytime we have a question CIA or so, often it's always you demonstrating your knowledge through solving the questions, solving problems, and so on. So this will be this is the how this style of question papers would also be there. Explain the significance of genetic engineering. I would never ask anything like that. Okay, keep that in mind. And there are some more things that we have to discuss. I think you know about star activity, some people who have studied, but otherwise we will see that. Say, for what is star activity? GAA-TTC. This is the sequence for EcoR1 and 5 prime, 3 prime. If 5 prime and 3 prime are not mentioned, the top strand from left to right is thought as 5 prime to 3 prime, and the bottom would have been the complementary of it and anti-parallel. Okay, so GAA-TTC. This is what EcoR1 recognizes. But because of some conditions, physiological conditions at which it is played, especially when you're doing in vitro, if there is a sequence like this, did I get it wrong? No. This is a sequence similar to the above sequence here, but it's not identical. It is similar in most of the positions except this position. In this position, there is a variation. If everything is optimal, EcoR1 would not be able to recognize and cleave it. It would cleave only here. But because of some conditions such as elevated pH or low ionic strength and so on, the enzyme might not work appropriately. It may have little bit change or drop in this specificity. It is, or its fidelity is reduced. because of which it may recognize this and then may mediate its cleavage here and here. So anytime we have something that is not accurately, where the enzyme is misfiring, those are referred to as, those parts are referred to as star activity. So whenever we are doing an experiment, we have to follow the exact conditions that are required. Usually the buffers are provided along with the enzymes, and then you have to use proper dilutions, and the precision with which we take the amounts of things that we take should also be very accurate. Otherwise, they may EcoR1 may recognize, cut something somewhat similar to that of the actual recognition site. The point of all genetic engineering is to be highly in control of what is happening, because there is nothing that we can see. We cut the DNA, we join DNA molecules, construct a recombinant DNA, and we try to express and so on. Unless we sequence this, we never know what if everything happened right or wrong. We cannot be sure if something is right or wrong. And that's why we need to be in control of things. That's why we should exercise a certain degree of paranoia in the way we perform the experiments. So so far we have discussed types, and we have also discussed the advantages of using type 2. We have discussed nomenclature, and we have discussed about 5' and 3' overhangs. I haven't discussed about isosygomers and neosygomers. And okay, isocardamers, they are, I will discuss these in the next class. Isocardamers are the ones, different enzymes recognizing different sequences but generating same overhangs. I will repeat what are isocardamers so that I won't want you to finish this from the table. Isocardamers are different enzymes, recognize different sequences. Recognition is different, but overhangs are similar, identical overhangs. So can you identify any from this table that is here? Can you identify isocardamers? Everything, they are different. All of them are different, so you don't have to worry about it. All of them are different sequence. They should recognize different sequences. I think if you find any of these enzymes here, you cannot call MBO1, DPN1, SAU3 as isocardamers. They would fall into, I think it is isosygomers. Let me get that checked, and I don't want to say what I'm confused with. So can you tell me any of the 2 restriction endonucleases that are isocardamers? They should recognize different sequence but generate identical overhangs. Anyone? Is there one actually? Well, these 2 will be isocardamers, right? But it doesn't make sense. Usually we'll talk about only about overhangs, not the ends. So let's check out. Yes, anybody has? You have to tell me 2 enzymes. Others, please type 2 enzymes so that we can then form. No, ALU is, yeah, we will not talk about the blunt ends. Let's see if there is any other. I found one. What is the axis of symmetry here? This one. What is its overhang? It has C, and if it is cut, it will generate CGG. And this is 5'. This will be 5'. This is 3'. This is 5' overhang, right? GGC, and here you have this, right? This is the overhang. One second. Is that right? Yes. So there is only one base. Oh, wait. I made a mistake. 1 second. It is only a 4 base cutter, so I need to keep that in mind. G, C. Yeah, this is the sequence. So there is the overhang would be. Oh, this one is not fine. Is there any other enzyme that generates 5' CG overhang? No. This one doesn't. This one doesn't because it generates only a C, single C. We want a overhang that is CG. Sriram has said MBO1 and South Korea should come as well. BGL2. Yes, Sriram is right. MBO1 will generate a GATC overhang and BGL2 will also generate a GTAC overhang. That's right, you're right. CLA1 and PVU1. CLA1 is C. PVU1 does not. This is generating a 5' overhang, and this is generating a 3' overhang. So they are not in the same. Not one, the axis is this, and the overhang would be GG. GGCC is the overhang. You have to find anything else that is going to generate GGCC. If you want to call not ones, you have to find the overhang alone. GGCC is the overhang. Is there? I don't see any other sequence that can produce that here. No, that's about it. I think that's only the one, CLA one. Ketira Balan, did I answer yours? Why CLA one and PVU do not match? Yeah?
Yes, sir, I understood.
That's good. Okay, then. I hope you all have understood what I wanted to say. In the next classes, we will try to construct the recombinant DNA and we will discuss about the many other enzymes that are useful. These are the tools that we should know. And once we finish that, we will know many more things. Okay, any other questions now, please? Just we'll have 5-10 minutes. Anybody knows about isocytomers? I think you can educate me. I forgot maybe. I got confused maybe. Neocytomers and isocytomers. To my understanding, different enzymes recognizing same and cutting same. But in my slide, I made a mistake maybe. So here it is recognizing same sequence, but cutting at different locations. These two are different. It says neocytomers. But I have written this. I think this is what I always believed, but because of this image that I chose, I'm a little bit confused. So here is SPH1, CGTACG, and BBU1. They should be italics. CGTACG. They cut in the same site. They are different enzymes isolated from different organisms. Recognition sequence is same, and cut site is also the same. Neocytomers are supposedly, they recognize same sequence, but cut at different points. So I will get back to you with this. And so don't carry anything into your mind at this point. If you remember, that's all right. If you can also verify, it will be helpful. Any other questions with assignment or anything? Are you all going to submit today? Hello? don't wait till the deadline, please. Make it happen. At the end, no excuse will be entertained. As you know, I already extended it by 3 days. So I would want that you do it today itself. Yes. Any other questions? Okay, I forgot to mention, I don't know where it is. In the next class, we will discuss about restriction mapping also. I will not discuss, I will actually pose the questions directly. I think you all have done before. Some of you were in my lab also, where we might have discussed. In molecular biology for bioengineering people, we did discuss these as a, in general, restriction mapping. Restriction mapping will come as a question in your exams too, because that's also something that will demand you to think a lot and visualize things. So restriction mapping is one thing that I want you to practice for the next class. We have, okay, we'll discuss that. Anything else? We have 5 more minutes. I want you to tell me something, any questions you had. Hello? I think I have to do some exercise. All of you can turn on your video now. Sleeping?
No, sir.
Okay. I'm just kidding. That's all right.